AP Bio students. It is Mrs. Shelton. We are going to be talking about one of my favorite topics ever, which is Chapter 7, Cellular Respiration. Hopefully in this chapter, what you start seeing is just how freaking cool biology actually is, just even down at the molecular level. So what we are talking about in Chapter 7, remember that you guys do have your You Must Knows that you need to make sure that you know before we come to our test next week. But in cellular respiration, hopefully at this point in your biology career, you definitely know what the reactants and products, if we were going to summarize cellular respiration, you know that we need to eat, you know that we need to breathe, and in that process, we are breaking down energy. So these are catabolic reactions that we are addressing and energy conversion reactions that we are addressing in going from something like glucose, C6H12O6, with the use of oxygen to now transfer that energy into ATP. So make sure that you are addressing the You Must Knows. And then with your notes, I'm going to go through on the Screencastify with your notes and make sure that we understand all of the major steps and some of the major events that actually happen in cellular respiration. So for the presentation, what we need to make sure that we are remembering is that with cellular respiration, let me hide this, presenter view, so we can actually see this better. This is not letting me do that. We'll just go on the small screen is what we will do. I'll put myself over here so we can talk about this. So first of all, when we do talk about aerobic respiration, we need to understand that respiration, this breakdown, this conversion of glucose, and we'll just go simplify it, glucose to ATP, is done by all things on this planet. So last chapter we talked about ATP and the work that ATP does, but we need to remember that when we look here at plants, a lot of times the misconception is that plants just perform photosynthesis and now animals do respiration. Please understand that every living thing on this planet does some form of respiration. So whether it's fermentation or it's aerobic respiration, this plant is going to be performing aerobic respiration. It does photosynthesis first, and then it uses that stored chemical energy of sugars and then uses those in order to convert it to ATP through aerobic respiration when needed. As an animal, animals are heterotrophs. We have to get our energy from some other source. So we only do aerobic respiration in our cells, but we still also do fermentation in some of our cells too, especially our skeletal muscle cells. We do a fermentation process, which is now not using oxygen when we are trying to transfer the energy from sugars into ATP. When we do look at the relationship between photosynthesis and cellular respiration, we do need to remember from chapter 1 when we talked about the fact that energy flows through living things. So energy, remember from chapter 6, we talked about law of conservation of energy. Energy can't be created or destroyed, it just changes form. So really what we're looking at is this change in form of energy, where light energy in the form of photons is used in the process of photosynthesis to now power this endergonic reaction, this anabolic activity of going from carbon dioxide and water to larger organic molecules. Now, a side product, so a waste product of photosynthesis, when water gets split in order to donate electrons and protons to electron transport and photosynthesis, oxygen is actually a waste product that is released. Well, with the mitochondria, in the inner membrane of the mitochondria, this oxygen actually gets used to power another electron transport and actually generate that proton concentration that now powers ATP synthase to convert the energy from organic molecules to ATP. Now, in the process, when we talk about converting organic molecules to ATP, the process that we're going to be addressing with cellular respiration is that we actually have to go through multiple steps in order to be able to strip out electrons and hydrogens from these organic molecules. Now, for simplicity's sake, we are going to be talking about sugar, but do realize that it's not just carbohydrates, but it's also lipids and proteins, so amino acids that can also be used in order to produce ATP and to generate energy that can get converted into the form of ATP. But we're going to talk about glucose, C6H12O6. in its combination with oxygen in order to then create the waste products of carbon dioxide and water and ATP. So this is just a summary equation, but why I'm showing you this summary equation is so we start looking at this as a redox reaction. When I talk about it in terms of a redox reaction, I need you to be aware of the electron transfers that are happening. The terms I absolutely need you guys to know is oxidized. Oxidized means that a molecule is losing electrons. In cellular respiration, sugar is going to lose electrons, and so it's oxidized. But also in this process, in the end step of oxidative phosphorylation, oxygen actually becomes reduced. Oxygen is that final electron acceptor in this process, and when oxygen accepts those electrons, it actually now bonds with hydrogen in order to form water. Oxygen is going to be reduced. So I need you to know those terms, oxidized and reduced, as we go through this process and all of the major steps and events that do happen in aerobic respiration. Now, because electrons are so important, and actually all these processes from glycolysis to pyruvate oxidation to the Krebs cycle, we are breaking molecules down and collecting electrons to be used in oxidative phosphorylation. Well, we need to have molecules that can actually transport those electrons to oxidative phosphorylation, or more specifically to the electron transport chain that is the inner membrane of the mitochondria. One of the major electron shuttles that you need to be aware of is NAD+. So NAD+ is an empty electron shuttle. You do not need to recognize this molecule and its structure, but the structure does kind of look familiar to you. It kind of looks like it's a nucleotide with an additional sugar, and you don't need to know that. However, NAD+ is what you definitely need to know. NAD+ has the ability to transport electrons. And then when it actually does pick up electrons and a proton, it actually now is NADH. So definitely know this relationship where NAD+ actually has the ability to become reduced. And so when it's in its reduced form, after it's collected electrons, it's now NADH. But then NADH will actually be oxidized at the beginning of the electron transport chain. And so then it becomes NAD+ again. So know that relationship between basically an empty electron uber and a full electron uber. It's just a shuttle for electrons going from one part of cellular respiration to the next part of cellular respiration. Now, in our book actually talks about aerobic respiration in 3 steps. We'll actually break this step down. So glycolysis being the first step. Glycolysis is actually an anaerobic process. It's part of fermentation, and it's also part of aerobic respiration. Glycolysis is this step of cellular respiration where basically glucose is getting split in half into 2 pyruvates. It does generate ATP, but it does not require any special structure. It happens in the cytosol of the cell, and every organism on this planet performs glycolysis to transfer energy from glucose to ATP. The next step that happens in aerobic respiration is pyruvate oxidation. This requires the mitochondria, and then followed by citric acid cycle. These processes, glycolysis, pyruvate oxidation, and citric acid cycle, are all involved in the oxidation of organic molecules. We are breaking down that glucose in order to collect electrons and protons. But then our last step here of oxidative phosphorylation, this is the final step that actually includes 2 parts to it. We can't have oxidative phosphorylation without electron transport and also chemiosmosis. This is where the electrons and the protons are going to get used to actually create and generate a tremendous amount of ATP. Now, just to illustrate the amount of ATP that is generated in this process, in glycolysis, my first step happens in the cytosol. They're showing you that I start with glucose, I end with pyruvate. I do collect some electrons here that are going to get shuttled to oxidative phosphorylation. Now I've got NADH. But per 1 glucose, glycolysis produces 2 ATPs at the substrate level. And by substrate level, I mean direct enzyme substrate collisions that would traditionally happen. Again, in glycolysis, all living things on this planet actually perform glycolysis. Now, if oxygen is present, now oxygen is not directly used in pyruvate oxidation or citric acid cycle, but oxygen has to be present for this process to proceed. for pyruvate to actually be transported into the mitochondria. And that's what I'm picturing here. So what's going to happen then is pyruvate oxidation is going to continue to break these pyruvate molecules apart. Now, glucose was a 6-carbon molecule. Pyruvate actually breaks it into 2 3-carbon molecules, is what I now have. And then what's going to happen is I'm going to oxidize pyruvate into acetyl-CoA. Well, acetyl-CoA is actually now 2 2-carbon molecules. So I actually get some carbon dioxide produced here. Those 2 2-carbon molecules are now going to enter the citric acid cycle, where the carbons and the oxygens are going to finally be released in the form of carbon dioxide. So I get carbon dioxide produced here in pyruvate oxidation. I get carbon dioxide produced here in citric acid cycle. But it's in the citric acid cycle, though, that I also generate 2 more ATPs at substrate level. In these processes, remember, along the way, I am collecting electrons and protons. In the citric acid cycle, I have 1 more electron shuttle that's called FADH2 is what I have. And so this is actually also going to be transporting those remaining electrons and protons, again, to the electron transport chain in oxidative phosphorylation. So in these steps, I can generate 2 ATPs per glucose in glycolysis. I can generate 2 ATPs per glucose in the citric acid cycle, also known as the Krebs cycle. But I could actually generate an oxidative phosphorylation when I now use the electrons and electron transport and the protons to power chemiosmosis. I can actually generate up to 34 more ATPs through this oxidative phosphorylation process. So this is also where we're going to spend a lot of our time, is focusing on the mechanisms by which oxidative phosphorylation actually occurs. Now, again, we have 2 different ways that ATP is regenerated, through substrate level and also oxidative phosphorylation. If I'm saying substrate level phosphorylation, what I mean is that I have direct enzyme substrate collisions that are generating my ATP, is what I need you guys to make sure that you're understanding. So this is just enzymes that are in the cytosol or enzymes that are in the matrix of the mitochondria. That's the middle portion of the mitochondria, that are now coming in contact with the substrates, which the substrates would be phosphate and ADP in order to create ATP. Oxidative phosphorylation does not have free enzymes in solution. It actually has embedded protein of ATP synthase in the inner membrane of the mitochondria. And we'll talk about that when we get there. First, though, let's talk about glycolysis. So what I need you guys to know about glycolysis is we start with a glucose molecule. Glucose is a 6-carbon molecule. And just like any chemical reaction, even though this is an exergonic catabolic reaction that is spontaneous, I still need an activation energy. So I actually have to have ATP present in order to initiate this process of starting to break glucose apart. So I input 2 ATPs, and in this process, what I actually end up producing is a total of 4 ATPs, which makes it sound like I lied to you and said only 2 ATPs are produced. Well, that is our net gain. So our net gain here is I have, with this, even though I made 4, I had to use 2. So I really only have 2 additional ATPs that were produced in the process. But also in this process of converting a glucose to 2 pyruvates, I also then collect electrons and protons. So I actually do get NADH produced, and I also get some protons produced too. But really it's the NADH that I'm most concerned with. But I still have organic molecules left in the form of pyruvate. A pyruvate is a 3-carbon molecule. So essentially what happens in glycolysis is glucose is split in half. And in that process, it took 2 ATPs to start the splitting process. I got 4 ATPs and 2 NADHs, but I really had a net gain of 2 ATPs per glucose in this process. So this is why I have a net down here of what my end products are. Now, that's oversimplifying things, but that's the level you need to know it for AP biology. Reality is glycolysis, this process that every living thing on this planet does, this is how it is tied evolutionary-wise to every single living thing, it is a 10-step process. So this is not just, oh, I take glucose and I split it in half into 2 pyruvates. That's all you need to know, though. But for future, if you do take a biochemistry class, you are going to have to know, I start with glucose, I add ATP, here's an intermediate, Here was an enzyme that actually that catalyzed this reaction. Here's another enzyme that catalyzed the next reaction. Here's where I get a split. And then from that split, then here's the continuation. It's a 10-step process. You do not need to know the 10-step process at this point in your biology career. If you take biochem as a third-year college chemistry class, that's when you get to learn all of these steps and the significance of each of these steps that are happening. All you need to know, I start with glucose, I end with 2 pyruvates. And in the process, I made 2 ATPs. And where this happens is in the cytosol of the cell, just that liquidy space inside of a cell, and that every living thing on this planet performs glycolysis. Now, if oxygen is present, so this is kind of the change that ends up happening. Once I have my pyruvates, and remember I have 2 pyruvates from 1 glucose, I have my 2 pyruvates. If oxygen is present, then aerobic respiration is going to continue in the mitochondria. If no oxygen is present, then fermentation occurs, and there's 2 different types of fermentation. We experience in our muscle cells, we experience lactic acid fermentation. That's what that pain is, that pain, no gain feeling in your muscles is lactic acid fermentation as a result of not delivering enough oxygen to your muscle fibers. Now, after glucose splits into pyruvate, I get my 2 ATPs, substrate level phosphorylation. Now what happens is, if oxygen's present, remember I have 2 pyruvates from glycolysis, I'm going to have 2 3-carbon molecules that start getting shuttled into the mitochondria. Well, what happens is these 3-carbon molecules, so 2, 3-carbon, 1, 2, 3, these are 3-carbon molecules, they are going to get oxidized. Remember, they're going to lose electrons. Now, how these pyruvates get oxidized, lose electrons, is there's enzymes that control this activity too, is each of the pyruvates is going to lose carbon and 2 oxygens in the form of carbon dioxide. So here's one of the waste products that is produced. I'm going to end up having per glucose 2 carbon dioxides that are released in this next step. I also collect electrons and hydrogens in the form of NADH. But then what I also now have is this 2-carbon molecule that is an acetyl group. So it's called acetyl, but it joins up with this enzyme called coenzyme A. And so when this enzyme coenzyme A groups with this 2-carbon molecule that's left, my only remaining part of my original glucoses. Remember, I've got 2 of these. Per glucose, I've got 2 of these events happening. Well, acetyl-CoA is this enzyme substrate complex that now joins the citric acid cycle. Now, the citric acid cycle is a very complex cycle. Again, you do not need to know every single step. Here's my citric acid cycle of every intermediate molecule that happens, but it's a cycle because it's a repeat. I start with this molecule called oxaloacetate or oxaloacetic acid. It's this initial molecule that has to be there to first connect with acetyl-CoA. And those 2 carbons are going to join in and form citrate. Now, when you see the ending ATE, citrate is also citric acid. So that means it's an acid, and that's where this cycle gets its name, is citric acid cycle. Now, its former name was called the Krebs cycle, named after the good doctor, Dr. Krebs, because that's what good scientists did back then, is they named things after themselves. We now refer to it as the citric acid cycle. College Board is going to call it citric acid cycle and Krebs cycle together, just in case you learned it one way or the other. Now, this 2-carbon molecule joins citrate. You do not need to know any of these intermediates, but what you do need to know is we are continuing to get rid of those carbons and oxygens, and see, this is where I'm seeing my carbon dioxide come off. And as carbon dioxide is coming off and being produced, I am getting more electrons and protons in the form of NADH. And also in this process, through substrate level phosphorylation, I'm generating ATP. Well, now, as I am also going back towards my starting molecule, I can also generate some electrons and protons in the form of FADH2. So FAD, FAD, is another electron shuttle that is able, just in the citric acid cycle, to pick up a few more electrons and protons. And then through magic or through science, right now you just need to know that ultimately what's going to happen is we are going to regenerate this original starting molecule, and we go back to the beginning. Again, you do not need to know all of these middle molecules that are there. This is the basics that you need to understand about the citric acid cycle. Acetyl-CoA comes in. This was a 2-carbon molecule. The coenzyme A goes and picks up more of the acetyl groups and brings them back. But I'm going to lose those 2 carbons in the form of carbon dioxide through a series of chemical reactions. What I'm also going to generate as I am pulling off these carbons and these oxygens and releasing the waste products of carbon dioxide, I am also collecting 3 NADHs per acetyl-CoA. I am also generating an ATP per acetyl-CoA, and I'm also generating an FADH2 per acetyl-CoA. That's what I need to know about the citric acid cycle. So in my pyruvate oxidation, I started with a pyruvate, and in pyruvate, I generated a carbon dioxide, I generated an NADH, and my ending molecule was acetyl-CoA. And then that acetyl-CoA then enters the citric acid cycle. And at the end of the citric acid cycle, I have no more organic molecule left from my original sugar. What I have at this point is the waste products of carbon dioxide, and I have the electrons and the protons that have now been saved in order to be able to deliver those to electron transport. So here's kind of an overview of where we're at right now. In glycolysis, happens in the cytosol of the cell, glucose is converted to 2 pyruvates. I went from a 6-carbon molecule to 2 3-carbon molecules. I generated 2 ATPs in this process. I generated NADH. That NADH is going to end up at oxidative phosphorylation. I also had pyruvate oxidation, where I went from 2 pyruvates to 2 acetyl-CoAs. This happens inside the mitochondrial matrix, is where this process happens. And in this process, I had carbon dioxide that was produced, but I also had NADHs, my electrons and protons, that were also harvested that can be used later. My acetyl-CoA enters the citric acid cycle. My citric acid cycle is going to generate 2 ATPs per glucose, 1 per acetyl-CoA. And also in this process, I'm going to generate 6 NADHs and 2 FADH2s. But this is also where I release the rest of my carbon in the form of carbon dioxide. So at this point, I have no more carbons left. I was able to get rid of them in the form of carbon dioxide, but now I have the electrons and the hydrogens remaining from my original organic molecule. So now we get to go to the cool part of oxidative phosphorylation, which there's a cool song that I will play for you. The whole point of oxidative phosphorylation is to power ATP synthase. This is an enzyme, and this is a freaking cool enzyme that's embedded in the inner mitochondrial membrane. And if we look at this enzyme, this model is actually making it look like it's a turbine, like it's a machine. It is. It is a turbine, like you would see in a hydroelectric dam, that as water flows through, it spins the generator, and that generator then is generating power. Well, this turbine called ATP synthase, this is an enzyme. It ends in ASE is the name. Anything ending in ASE is always an enzyme, and its job is to provide ATP synthesis. It is going to be ADP plus an inorganic phosphate. It is going to power that production of phosphorylating ADP and turning it into ATP. But how it does it is it generates its power from protons flowing through. If I have a high hydrogen concentration, high proton concentration in between the outer and the inner membrane, this is referred to as the intermembrane space. Well, protons are going to move from high concentration toward low concentration, from that inner membrane space back into the inner mitochondrial matrix, that middle of the mitochondria. Well, as those protons flow, it actually literally turns this turbine. And what's happening is it turns this turbine, that enzyme is grabbing ADP and phosphates and just slapping those suckers together. So that is how I get ATP synthase to power. But let's back it up a little bit and understand how those electrons and protons got to this place. What I need you guys to first of all understand is membrane proteins in the inner mitochondrial membrane. They are actually organized from higher energy levels to lower energy levels. So what's going to happen is in that inner mitochondrial membrane, all these integral proteins that are embedded here. Well, NADH is going to actually pass off its electrons to this first protein. This is actually just a series of oxidation reduction reactions that end up happening. These electrons then are going to move from a higher energy state to a lower energy state, to a lower energy state, to a lower energy state, all the way down. So they are moving from higher energy state to lower energy state. As they're moving, that's electricity. That's what electricity is the flow of electrons. And so we have this little electrical current that's moving down through these membrane proteins. And as they move down these membrane proteins, well, there's these other integral proteins that are proton pumps. And what they end up doing is powering these proton pumps to generate a high concentration of protons as these electrons move through. Now, FADH2, it's also going to drop off its electrons, but it doesn't drop it off at as high of an energy state. The electrons are dropped off at a little bit lower energy state, so they still generate some power through proton pumps, but not as much. But these electrons are always going to move from higher energy state to lower energy state. But what makes them continue to move and continue to flow is oxygen has to be present at the end of this electron transport chain. Oxygen is the most electronegative. Oxygen is the final electron acceptor at the end of electron transport chain. And when oxygen then binds with these electrons, when oxygen gets reduced, oxygen is actually going to be converted into water. So this is where my product of water is established. This is also where my reactant of oxygen technically has to be used in the chemical process, is being that final electron acceptor of electron transport chain. So in another schematic, let's just kind of talk about how both of these work together. So here's a diagram from your book that you are definitely going to need to make sure that you can explain. What I am seeing here is the inner membrane of the mitochondria. Remember, mitochondria is a double membrane structure. The inner membrane has all of these membrane proteins. Now, for simplicity's sake, they have taken out all of these small little membrane proteins just to make sure that you can see kind of where does NADH and FADH2, they are dropping off their electrons and their protons here in the matrix. On matrix side is where they are dropping off their electrons and their protons. Electrons that are moving from a higher energy state protein towards the lowest energy state possible at the end, which is oxygen. So this yellow line here is showing you the flow of electrons through the electron transport chain. And that's why it's called electron transport chain. It's a chain reaction of oxidation, reduction, all the way through these membrane proteins, which is moving of electrons is electricity. As the electrons are moving from higher energy state to lowest energy state possible, which is now in water. Well, what they're moving through are these proton pumps. These proton pumps gain their power, and if they're proton pumps, they pump protons. Well, the protons actually were released here in this matrix of the mitochondria. Well, now that electrons are flowing through them, generating power through them, well, that gives enough energy for these proton pumps to move those protons into this intermembrane space. So what's going to happen, kind of like a hydroelectric dam, I build up a high concentration behind this wall of protons. So I end up having an incredibly high concentration of protons back here. That's the purpose of electron transport chain. This flow of electrons powers proton pumps that generates this high proton concentration in the inner membrane space. So the second step of oxidative phosphorylation is now chemiosmosis. Now, when I say chemiosmosis, you should be thinking of osmosis, diffusion of water. But I'm not going to have diffusion of water. I'm going to have chemiosmosis, diffusion of chemicals. What chemiosmosis refers to is this diffusion of protons from higher concentration area to lower concentration area through the enzyme ATP synthase. So if I don't have electron transport chain, I will not have chemiosmosis. Can't have chemiosmosis without electron transport chain. So I collectively refer to them as oxidative phosphorylation. Now, this phosphorylation was the addition of a phosphate to ADP. That's where I get that term phosphorylation. It means sticking a phosphate on another molecule. And in this case, I am sticking it on ADP to regenerate ATP. It's oxidative phosphorylation because ultimately what powered this is the oxidation of glucose. I oxidize glucose to get those electrons and those protons in order to power this process, is what I had to do. So again, here's my electron transport, just in simple form, showing you that NADH and FADH2 are dropping the electrons off. This again is the matrix side. This is the inner membrane space. So the outer membrane would be up here. Here's my inner membrane of the mitochondria. Electrons are going to flow from higher energy state to lower energy state, with oxygen being that lowest final electron acceptor, forming water. Remember that from this picture, it is a little misleading from the picture, because if we look at it from energy states, these membrane proteins pass the electrons down from highest energy state down to lowest energy state. Again, as they are moving, they are powering these proton pumps. And that's what I'm seeing here are these proton pumps that are now actively transporting protons into this inner membrane space. And now that I have a high concentration of protons in this inner membrane space, remember, charged particles can't diffuse back through a lipid bilayer, but protons are then given a space to flow through this enzyme ATP synthase. And when protons move then from higher concentration down to lower concentration, this is what is now powering ATP synthase. That's an integral protein, but it's still an enzyme that is catalyzing this reaction. It's getting its energy from this flow of protons from high concentration back to low concentration, which is in the matrix. So with this, remember that shape of that particular ATP synthase, it really is a turbine that is literally spinning, grabbing ADP and phosphates and slamming them together. But it's only going to spin and crank out ATP if I get protons that flow. I am only going to have protons that flow if I have oxygen down here at the end of electron transport chain. Now, if I did not have oxygen here at the end of electron transport chain, well, now electrons won't flow. If electrons don't flow, well, protons don't pump, and they don't create this high concentration gradient. But like I also told you, is this is the only place that oxygen actually gets used. So why are the other steps? Why is citric acid cycle dependent on oxygen? Why is then pyruvate oxidation dependent on oxygen? Well, it has everything to do with these electron shuttles. If I stop electron transport, then FADH2 has nowhere to drop off its electrons. NADH has nowhere to drop off its electrons. And if I can't drop off my electrons, then there's no NAD plus or FAD to pick up electrons here. And so these systems actually stop as well. So I have to be able to empty out my Ubers or empty out my taxis so taxis can go pick up more passengers and bring them back. So if I don't have oxygen here, there is nowhere for NADH or FADH2 to empty themselves out and go back and pick up more electrons. So all of these systems shut down if oxygen is not present as the final electron acceptor in electron transport. Now, this is also why in anaerobic organisms, when oxygen is not present, organisms and some cells, so not every cell in our body is going to do fermentation, but after glycolysis, because remember, glycolysis produced NADH. Well, if I don't have anywhere to now drop off those electrons, I got to get rid of them somehow. So this is why fermentation occurs. Fermentation is actually a process that actually allows the cell to utilize the energy that's in the form of electrons and protons to convert pyruvate to something else. And that's really all that's happening in these schematics. Glycolysis still happens. Glycolysis generates 2 ATP. Glycolysis generates 2 NADHs. But if these NADHs don't empty themselves out, glycolysis is going to stop too. I'm not going to have any more taxis that can pick up more electrons, and glycolysis shuts down. So in order to free up NAD+, different cells are going to do 2 different types of fermentation. All they're doing is turning pyruvate into some other end product using the electrons and the protons. In this case, with alcohol fermentation, pyruvate We also cleave off of carbon dioxide. So this is what makes your bread fluffy, is when you use a yeast that does alcohol fermentation. No, you are not going to get drunk from bread. You might go into a food coma from drinking too much or from eating too much bread. But carbon dioxide is released. That's what makes the little air bubbles in the air pockets in your baking goods. But alcohol then is the end product. With heat, alcohol actually evaporates out of your bread products is what ends up happening. But that's a fermentation process. And also what makes your yogurt tangy or your skeletal muscle fibers sting is actually now a pyruvate conversion to lactic acid, to lactate. But again, it's a fermentation process that's utilizing the electrons and the protons from NADH, and it's freeing up NAD+. Because if I did not have NAD+, glycolysis would stop, is what would end up happening. So understand the role of fermentation and its effect on NAD+ and reestablishing that. Now, what I also lastly need you to know, because we talked about cellular respiration from a glucose perspective, but what I also need you to know is that it's not just carbohydrates, but it's amino acids, it's different parts of lipids, the glycerol, fatty acids. These can also be oxidized in order to help power oxidative phosphorylation. So just be aware that proteins, carbs, and lipids can all be used as an energy source in our body and in all living things. However, nucleic acids, your body would prefer to use nucleic acids to make things like NAD+, make ATP, make DNA, make RNA. So those we do not break down as an energy source. So in the last review, as we take a look at this diagram, remember that we start with glucose. Glucose is broken down in glycolysis to 2 3-carbon molecules of pyruvate. We generate 2 ATPs through substrate level phosphorylation. We also generate 2 NADHs. Remember, NADH is carrying electrons and protons. Remember that glycolysis happens out in the cytosol. So since there's no special structure that is necessary, all living things on this planet perform glycolysis. That's a very important thing to make sure that you are remembering. But now if oxygen is present and we have a mitochondria, pyruvate is actually going to be oxidized into 2 acetyl-CoA molecules in the mitochondria. Those 2 acetyl-CoA molecules, remember in acetyl-CoA, each of those are 2 carbon molecules. So I actually produce carbon dioxide in this step as a waste product, and I also am collecting 2 NADHs. My 2 acetyl-CoAs, those 2 carbon molecules enter the citric acid cycle and are fully oxidized through the citric acid cycle. I also generate 2 ATPs through substrate level phosphorylation again. These events are both happening inside the mitochondrial matrix, is where they're happening. Carbon dioxide is also a waste product in the citric acid cycle. Remember, carbon dioxide and oxygen, they easily diffuse in and out of every membrane, so it diffuses out and we breathe out that carbon dioxide, is what ends up happening. But I collected a lot of electrons and hydrogens in those, and then we use those electrons in electron transport chain to actually then pump the protons into this inner membrane space between the inner and the outer membrane. So here's my inner membrane, outer membrane. That's why it's actually colored orange here. It's showing you this high proton concentration. But my inner membrane is where ATP synthase is. My inner membrane is where electron transport chain takes place. And so then through chemiosmosis, I'm going to have the diffusion of protons back through ATP synthase, generating, in this schematic, they're showing you from 26 to 28 ATPs. That's realistic. But just know, in ideal situations, if everything worked perfect, there was no energy requirement used for any other system, we could actually generate 34 ATPs in this process. So per glucose, we can get anywhere from 30 to 38 ATPs. But these ATPs here are all generated via oxidative phosphorylation. There's a difference between oxidative phosphorylation versus substrate level phosphorylations that end up happening. College Board will never ask you exactly how many ATPs are made in oxidative phosphorylation. You should just know a whole lot more than through substrate level phosphorylation and glycolysis in the citric acid cycle. Okay? So with that being said, I definitely need you guys to review, ask questions, understand these steps of cellular respiration. But most important, here's just my what I think is just so freaking cool is just this enzyme ATP synthase. Now, last question. Do you think any prokaryotes have ATP synthase? And the answer is yes, they do. Yes, they do. There are some prokaryotes that actually have membrane proteins that transport electrons. They actually will generate a high proton concentration outside. Protons will actually diffuse through this. They just don't have a double membrane structure, but they still actually perform an aerobic respiration. They just have these membranes. These membranes and the membrane proteins are their outer plasma membrane. So don't just think that oxidative phosphorylation is a eukaryote thing. We've talked about it only in the mitochondria, but there's limited oxidative phosphorylation that does happen in some prokaryotes that end up having these membrane proteins that can do electron transport, and they also have ATP synthase. So the fact that we actually have mitochondria then that are a double membrane structure, this is further evidence of endosymbiont theory as well. So with that being said, that was a lot of information for y'all today, but break it down. That's why we're breaking it down in class, is why we are doing that. But ask lots of questions, take it section by section as we go through. Stop me at different sections as well in order to get the main points of each of those sections. But hopefully you enjoyed cellular respiration, all of the chapter all at one time, and I will see you guys in class tomorrow.